So again, thank you guys for coming. This week we're gonna do week 1 topic review. And so that's all like the amino acids, protein structure, protein folding, things like that. Oh, I'm Tina, by the way. And then
I'm Michaela.
Okay. So starting off with amino acids and levels of protein structure. And obviously, if you guys have any questions along the way, feel free to like stop us at any time. You can just raise a hand if you're in person, or if you're on Zoom, you can like type it in the chat or like unmute yourself. Anything is okay. We're a small group. Okay. So starting off with amino acid structure, an amino acid has essentially 3 parts. You have your amino group, which is the NH2 in this case, the carboxyl group, so C double bond O with the OH, and then your R group, which is the part that is different for every amino acid. That's like the special part of an amino acid that gives it its characteristic. Technically, like the 4th part is just the hydrogen bond connected to the central carbon. Sometimes you'll hear Laura talk about the central carbon that is like a alpha carbon. So that's just like another terminology to be familiar with or just have heard of, so that you're not caught off guard if that ever comes up. Another aspect of amino acids in general is the formation of peptide bonds. So when amino acids come together, that's called a peptide bond, that they're joined through a hydrolysis reaction. And then the bond that forms between the 2 amino acids is a peptide bond. So specifically what occurs is you have the OH from the carboxyl group of 1 amino acid, and then the H of the amino group from another amino acid. Those 2 break off into a water molecule. So that's why it's a hydrolysis reaction or a dehydration reaction, because the water is leaving, as you see over here. And then the carbon and the nitrogen bond together. And that bond between the carbon and the nitrogen is a peptide bond. What's important to know is that a peptide bond is a covalent bond, and we're going to talk more about like all the different types of bonds that occur in proteins, but a covalent bond in the case of biology is very strong. So peptide bonds between amino acids are very strong. It's hard to break them. It's not something that's easily like changeable. So that's just something to keep in mind. Oh, and then another point is that the peptide bond formation is catalyzed by the ribosome. A ribosome is an enzyme, but the enzymatic part of a ribosome, since ribosomes are made up of proteins and RNA, the rRNA of the large subunit of a ribosome is the enzymatic or like catalytic portion. So that's the part that's actually, what's it called? That's the part that's actually like catalyzing and helping out to form this reaction. So I think that is one part of your problem set. So keep note of that. That might help you out. Yeah.
So you said the RNA is what helps catalyze, and then the ribosome does it like binding it?
So the question was like specifically what the ribosome does versus like what the rRNA does, kind of. So the ribosome is like an enzyme. Like a ribosome is like one big thing in the cell. And so ribosomes are made up of proteins and rRNA. And so it's not the entire ribosome that acts as an enzyme. It's specifically like the rRNA part of a ribosome that does like the catalyzing. Yeah. So if you want to think like you have a ribosome is a type of ribozyme. So it's an enzyme. And then a ribosome is made up of RNA and protein. But the protein's just like there that is like making up the ribosome, but the rRNA is like actually catalyzing all the reactions to make the amino acids chains. Yeah, does that make sense? Okay. Okay. So when a bunch of amino acids come together via their peptide bonds, that's called a polypeptide. So poly is like multiple, so you have multiple peptides coming together, hence polypeptide chain. to explain a little bit more about the structure of it so that you guys start getting used to terminology of amino acids and proteins in this class, you have an N terminus and you have a C terminus. Sort of easy to remember once you kind of get a hand of why it's called such. The N terminus is the side of the amino group. So amino has a nitrogen, hence the N terminus. The C terminus is the carboxyl side. So it has a C double bond O with like the OH group. So that's why that's the C terminus. You always start with the N terminus and then you end with the C terminus. So if you had to like label all your amino acids, the N terminus would be number 1, ending at like, I don't know, 559 for C terminus. So that's what you would label. So if you ever had a question that was like, oh, what if you cut it off at like the beginning of an amino acid, or sorry, if you cut it off at the beginning of a polypeptide chain, which terminus would you be closer to? It would be the N terminus because that's at like the beginning of the peptide chain. Okay. Yeah. No, this one?
Yeah. So on the right, it says that like the negative dipole is on the oxygen, but on the left, is it on the hydrogen? For the N terminus?
For this one?
Yeah.
So there's different, not different, like an amino acid has a basic structure, and that was like the slide that I showed before. But when you're in like a body versus when you're outside of a body, like different types of pHs can cause an amino acid to either have like a positive charge, a negative charge, or be neutral. So when it's neutral, it's like this is an OH and then this is NH2. But when it is charged, the amino group becomes an NH3, so that makes it positive. And then the oxygen loses its H, so then it becomes negative. So it just, you don't need to worry too much about it. But it is like something that happens because of different pHs and depending on where it is. Yeah. Does that make sense? Okay. Yeah. Okay. And then last thing would just be that peptide bonds, even though they are single bonds, I said before that they're really strong. And the reason is because of resonance, all the electrons that are moving around. So single bonds are typically considered like the longest bonds, but peptide bonds are like shorter types of single bonds. And that's sort of that's like one aspect of what makes it a stronger single bond than usual. Okay. Which one? Oh, we're not there. The protein structure level. So we're going to go through like the different types of levels. There are 4. Your primary structure, so like the first structure, if you want to think of it that way, is a sequence of amino acids. That's the plain definition of a primary structure. And so if you ever got a question about like what levels of protein structures would be changed if you took out one of the amino acids, primary structure would definitely be changed, because if you started off with like A, B, C and you took out B, that's not the same sequence of amino acids that you had in the beginning. So your primary structure is changed. Those are like kind of common questions that you might get to. So in the primary structure, it's just a sequence of amino acids. So all of your peptide bonds are linking the amino acids together. Again, peptide bonds are covalent, and covalent bonds are very strong, or the strongest in this class for this class purposes. So because they're so strong, they do require a lot of energy to break. Usually you need like the help of an enzyme in order to break it. They can't just be broken by like moving around too much or whatever other typical way to break bonds would be. So then you have secondary structure. Secondary structure, you're still talking about the backbone, or like the main part of the amino acid that does not involve the R groups yet. Secondary structure, you can have two different types of like shapes that you would find. One is an alpha helix, the other is a beta sheet. The way that they're formed is by hydrogen bonding with the backbone atoms. So again, to emphasize, the R groups are not being used at all to form secondary structure. It's all the backbone. So it's all the amino groups and the carboxyl groups interacting with each other that are forming these helical structure and like sheet structures. The reason why they're forming is because the backbone has dipoles. So you have temporary partial charges on the NH in the amino groups, and you have temporary partial charges on the OH bond in the carboxyl group. When you have partial charges, your electrons are moving around a lot. So then that causes hydrogen bonds to be able to form together. So all these like little dotted lines between the backbones, those are hydrogen bonds that are formed because of partial charges on the backbone. Does that make sense so far? If there's any questions, feel free to like stop me or slow me down. Yes, and then so hydrogen bonds, so far we were talking about covalent bonds with like peptide bonds. Hydrogen bonds are not covalent, so they are not as strong as peptide bonds. And then to go into like a little bit more specifically, alpha helices, all of the R groups are faced outwards. So you're not going to find your R groups like inside the helical shape. It's going to be facing outward, and it's every 4 amino acids that get paired together. It's kind of hard to see in this picture, but like this bond and this bond, these are like 4 amino acids apart. So that's good to know because that's what causes the helical structure to form. And then for beta sheets, your R groups are alternating up and down. They're never going to be right side by side. So if you can see like this one's up, this one's down, this one's up, this one's down. And that's to avoid what's called steric hindrance, which is basically when you have big things next to each other, they don't want to be next to each other. They're going to repel. So just to avoid having that hassle, they alternate up and down so that each R group has enough space for itself. Moving on to tertiary structure, your tertiary structure is, or like the third structure, that's when you start getting protein shape and protein folding because your R groups are being involved now. So specifically, it's a 3D structure where a single polypeptide chain is going to be folding in on itself based on the different types of amino acids it has, because each amino acid has its own characteristics, and that's what's going to cause the different chemistry bonds to form. So there's the main bonds that you have to be aware of for this class in regards to proteins are hydrophobic interactions or van der Waals, ionic bonds, hydrogen bonds, and disulfide bonds. And then we'll talk a little bit more about each one of those in the next slide. Just kidding, not the next slide, but we will talk about them soon. After tertiary is the quaternary structure, and quaternary structure is when you have multiple polypeptide chains. So we talked about structure, like primary structure, secondary structure, tertiary structure, that would be involved for just like this one purple part. But the minute you get like multiple polypeptides together, where this one's in tertiary structure, this one's in tertiary structure, this one's in tertiary structure, if they all come together, now it's a quaternary structure. So you have 2 or more subunits that are interacting with their backbones and R groups. And then the same bonds that apply to tertiary, so like hydrophobic, hydrogen, ionic, all those apply to quaternary as well. Okay. So just to emphasize, because a lot of students in the beginning get confused about tertiary and quaternary and the difference between the two. A tertiary structure is a single polypeptide chain and it folds on itself, whereas quaternary structure has multiple polypeptide chains, and then those multiple ones can bind with each other or bond with each other. What bonds? So the bonds that hold subunits together are the same bonds that can hold a tertiary structure together. So it can be your hydrogen bonds, your hydrophobic interactions, your covalent bonds, disulfide bonds, things like that. Anything that holds a tertiary structure, like one chain together, those bonds can also be used to hold different subunits together. Does that answer your question?
Yeah, thank you.
No problem.
It's still you. Would you want me to?
You know, go for it. Yeah.
Okay.
Oh, yes.
Okay.
I can't.
Okay, so for domains versus subunits, a lot of people get this confused quite easily. A domain is an independently folded section in a single subunit. So there's like multiple domains, there can be multiple domains in a subunit. And normally, we're going to go over this later, but like for an example, an SH2 domain is found in multiple proteins, but that one domain serves the same function. So a domain, even though like how proteins, the way they fold determines their function. So like that one folded section can be the same in each. I don't know if that confused anyone. Okay. And then a subunit is the two or more separate chains that can orient in 3D space to give a quaternary structure. So it's a single polypeptide in a protein that makes up multiple peptides. Are there any questions?
Do you want to explain
Yeah.
what a domain is again?
So a domain is a single chain that can form a local 3D structure. So it's an independently folded section in a single subunit. So in that folded sections, the R groups only interact with each other and not like the R groups in a separate domain, but on the same subunit. Does that answer your question? Okay.
Thanks.
Do you want? I don't know. What
Yeah.
Mhm.
Is the domain like still like, is it like different polypeptides? Or like outside?
No. So it's just one polypeptide. If on this picture, the domain, the example of a domain is on the left. Yeah, the red and green one. So in that picture or in that protein, it's one single protein where it's like one long polypeptide chain, but it has two different domains. And you can kind of tell because like the green side is one domain and then the red side is another domain. But even if it's not like color coordinated, because sometimes on exams it might not be color coordinated, you can kind of tell because like one side is clumped together and the other side is clumped together in its own. So all the green R groups are going to be interacting with each other and forming bonds with each other. Whereas on the red side, all of those R groups are going to be interacting with each other, but it's all one chain.
Oh, all one chain,
Yeah.
like different, like, okay.
Yeah.
Okay.
It's a little bit like you have to wrap your head around it, but the more you kind of like repeat it to yourself and look at pictures, it gets better. If at the end you still like want some clarifications, we can go over it more.
I was gonna ask one more thing.
No, go for it.
Is
Yeah.
it like depending on like if it's like hydrophilic or hydrophilic type thing? Or?
Do you want to answer?
Not necessarily. The reason that they fold independently, like within the subunit, is just because that one domain is going to serve one function and then the other is going to do something else, but all within the same protein.
Yeah. Hydrophobic and hydrophilic is kind of like the chemistry behind why things fold a certain way, but like function wise, it's like they have their own functions in a cell. Like each domain does different things.
Okay, so
Yeah.
different functions, but same protein.
Same protein strand, yeah. Let's skip the train.
Yeah. Okay. So then here's just where we're going to go over the different ways the R groups interact with each other. So for polar charged R groups, they're going to make ionic bonds. So off to the chart, the green and the purple here, those are the charged ones. Yeah, they're electrostatic interactions. And then for polar uncharged R groups, they're going to make hydrogen bonds. And the reason they can make hydrogen bonds is because in what makes an R group polar is that there's an unequal sharing of electrons. So that's what creates the differences in electronegativity. So like, for an example, in a hydroxyl group, the oxygen is going to have like a partially negative dipole on it. So then that's what allows it to interact with a partially positive dipole on a hydrogen of like another R group. So like, for example, on serine, the oxygen can interact and create a hydrogen bond with the hydrogen on threonine. And then for nonpolar R groups, they're hydrophobic interactions and they're weaker. They're sometimes called van der Waals or London dispersion forces. And the reason why they're nonpolar is because they're similar in electronegativity. Like C, carbon and hydrogen have similar electronegativity values, so there's no dipoles being created there. And then the hydrophobic effect is just the tendency of hydrophobic nonpolar amino acids to bind with each other to avoid water. And then I don't know if it's on this problem set or it may show up on an exam, but not all of the amino acids fit perfectly in one category. For example, tyrosine can sometimes be considered non-polar because of its benzene ring. So it has a lot of carbon-carbon and carbon-hydrogen interactions. Are there any questions on this slide? It's the chat. Okay. And then here's just an image showing how the proteins fold. So in the green, it's the non-polar R groups. So you can see how they're on the image on the right, they are clumping together to avoid interacting with the water, except they don't necessarily always clump together in the inside. It depends the environment that they're in. So I think like on the problem set, it's talking about in a membrane. So just be aware that not necessarily they're always going to clump in the middle.
Something to just add, kind of what Michaela was saying about like not all amino acids are grouped equally. For example, if you have like a really, really hydrophobic chain that's like, it has like a ring and it has a bunch of C's and H's, but then you have like alanine, which is just a CH3, then if you had to pick between the two of like which one's gonna hide away from water and then which one would be like okay with water, you would choose alanine to like be on the outside and then the really, really hydrophobic one to be on the inside. Even though they're both non-polar, it's kind of like which one is the lesser of the two evils type of thing. So you might get asked those type of questions and you just have to compare like which one's more hydrophobic, which one's less hydrophobic, and kind of use the knowledge that you know about like the basic things and then apply it.
And I think in one of the lecture videos, Laura also gives a chart or diagram ranking from least or most non-polar, I guess, to then most polar. So if you're not quite sure, you can look and then try and like figure out why that is and use that to help your explanation. Are there any questions? Okay.
Sorry. That's a weird thing.
Okay. So disulfide bonds, these are the only covalent bond that you'll find in tertiary and quaternary structure, and they can only occur between cysteine molecules, the sulfurs of the cysteines. So they're also going to be, since they're covalent, they are going to be stronger than the hydrogen bonds and the van der Waals. No.
They also, like I've seen this in the past, methionine has a sulfur, but that's not going to create a sulfide bond or like a sulfide bridge because it's not like an SH at the end of the amino acid. So unless you see cysteine, you're not going to have a disulfide bond. Yeah.
Okay, and then here's just a summary of all the bonds that occur in each structure. So the covalent bonds are peptide bonds, which are in the primary structure, and then disulfide bridges are only going to be found in tertiary and quaternary. And then for non-covalent, the ionic bonds between the polar charged R groups, hydrogen bonds, polar and charged, and van der Waals nonpolar. I also don't know if she talks about it, but you can have ion dipole interactions. So it would be between a polar charged R group and then a polar uncharged R group. And then,
Ion dipole.
yeah.
Yeah. So it's like
I could go back.
a dipole is when you have like a partial charge. So polar groups usually have partial charges, whereas like charged groups are permanently charged. So if you had like something from the green section and then something from the blue section, they can still bond, but that would just be called like an ion dipole. Sometimes she like tricks you with like different questions that kind of got covered, but kind of didn't get covered. So
Yeah. And then ion dipole, I guess, would just be stronger than hydrogen, but weaker than ionic here. Okay. And then for protein modeling, there's the ball and stick / wireframe model. The balls represent each atom and then the sticks are the bonds. So every atom and bond in the protein is modeled in this. So sometimes most of the time it's hard to discern between backbone and R group. And then wireframe just doesn't have the balls, but it has all the sticks, so it shows all the bonds. And then in the next slide, there's going to be pictures of each. And then the space fill model shows every atom and the space they take up. So this model is really good for looking at the overall shape of the protein, but it's hard to see the backbone. And then in the ribbon model, it highlights the secondary structure. So it's going to show the alpha helices and beta sheets. But the downside of this is that you cannot see the R groups. And then in the backbone, it only shows the primary structure. It only shows the path of the polypeptide backbone and doesn't show R groups. So it's like the skeleton outline of a protein. And I don't think it's on this problem set, but it may be on an exam in the future. You can use more than one model to show something. So for example, if you want to highlight the secondary structure, but also the R groups, you could combine like the ribbon model and then use the balls from the ball and stick to highlight the R groups. And then we kind of went over it here, but just to try and come up with pros and cons of each. And then here are just the images of all the models. Wireframe isn't on here, but it's just ball and stick minus the atoms.
Does anyone have any questions that, like content wise first, any questions? Yeah, if she ever gives like right, you guys still got them on. Yeah, if she ever asks any questions, you don't have to memorize them. You'll always get like that colorful, like blue, green, whatever other color there was, that graph will always be given.
Let me, where is it? Okay, so the ribbon model is going to highlight the secondary structure, whereas the backbone model is highlighting the primary structure. So, like the ribbon, you would see the alpha helices and beta sheets. Both you don't see the R group, but ribbon's just going a step, a step further than showing the secondary.
Yeah, if you want to show it the next slide. Yeah.
Yeah.
So like in the ribbon model, you can see like the twirls and you can see like the little flat areas. Those are your secondary structures. But in the backbone model, like all you see is literally the generic shape of the primary backbone, or the backbone of the amino acids, which is why it's called backbone model. Does that clarify that? Okay, cool. Any other generic like content questions?
I have a question about the beta sheet. So on a beta sheet, would like hydrophobic amino acids, because I know they're arranged in like opposing, like opposite directions, the R groups, would the hydrophobic ones be like on the inside kind of the beta sheet? Like how would that work? Like for hydrophobic amino acids and hydrophilic ones?
I think that's a valid question. It's just more like when you think of secondary structures, the things that are bonding are your backbone structures, right? So like the amino groups and the carboxyl groups, and then your R groups that are interchanging. Once it gets into tertiary structure, that's when like whether or not they're hydrophobic or hydrophilic or more or less, that's when that gets taken into consideration. And then within the beta sheet structure, it'll fold even more and bond accordingly so that the R groups that like want to be away from water will move away, and then so on and so forth.
Okay, yeah, that makes sense. Thank you.
If no one else has like content questions, we'll stop the recording. And then if you guys have like specific problem set questions, we can answer that for a little bit before the time stops. Yeah, I think you just click it.
Oh.
Well.